So in library preparation, what we do here is we have already extracted RNA or DNA, and what we do next is we will fragment that, and then we will prepare it to be placed into the sequencers for sequencing. So all those steps before putting it into the sequencer, we can call them as library preparations. So what we do here is we generate appropriate size fragments, and then we add primers or adapters, and in the end we screen for those fragments which are good for sequencing. So if we look into the workflow, obviously the library preparation is the second phase. Here is an elaboration of library preparations. So we have this large amount of DNA. In fragmentation, we shear them with some sonications or with sound waves, or we can use some chemicals to do that. And in the end, we see our DNA has been fragmented. Then after those fragments have been achieved, we add adapters to it. So adapters are the molecules that will help attach these fragments into the places like the flow cells where the sequencing reactions are performed, or in case of 454 machines, we will see the beads on which it attaches the DNA. So here we have the adapters which are ligated. So in addition to that, these adapters, sometimes we also add some additional things like barcodes. Barcodes are special sequences. We put them there in order to distinguish different kinds of materials from one another. So we can place some known nucleotide fragments, and we can help identify our DNA from that. So we might not need barcodes, they are not compulsory. Primers are there in order to do the amplification. So since we know that this DNA sequencing starts from the primers, so primers are also ligated. But in the modern techniques, we have observed that adapters, they have also been kind of manipulated, where we can take the role of the primers as well. So in screening, what we do here is we select those fragments that are having the appropriate size. That depends upon the technology and the length of sequence it requires for sequencing. So it should be up to that length, and obviously it should have all those important ingredients like primers or barcodes or adapters. So good fragments are the ones which are selected on the basis of presence of these important components. We prepare templates since we know that our sequences are double-stranded. So here in this small animation, we can see again the attachment of adapters, right? And the adapters are attached to these two fragments, so they are selected. Rest of them are discarded, and then we take these fragments and we make them single-stranded. So since we know that replication is a single-stranded process, so same is used in sequencing. So having a single-stranded templates is an important requirement in sequencing. In RNA library preparations, there is one additional step. So since we cannot take the RNA into our sequencers, we need to convert it into the DNA. So that DNA is called as the complementary or cDNA. So there is an additional step. So we fragment RNA and we convert that into cDNA, and then this cDNA is then used and it is put into the sequencer as a DNA library. In 454 sequencing, there is a small difference in the adapters, that one of the adapter contains biotin that helps attaching those fragments to the streptavidin coated beads. So those beads are used as a amplification and sequencing stations. We will look into them later on. In paired end sequencing, which is a special technique in which we skip some parts of the genome and we take the neighboring parts of that region, and we sequence them. So that's kind of paired end sequencing. So it has different applications. What we do here is we fragment DNA and attach adapters. Then we ligate it in a circle, and then we want to skip most part of this circle, and then we just take the DNA which is around those regions. So we cut it with the restriction enzyme, So then there is one fragment over there that contains these adapters. So if we take them, we don't need to take these rest of them. So in this way, this kind of technique is called as the parent sequencing. We can select those fragments which are containing those adapters, and then we can attach the sequencing adapters to these selected segments, and then we present them for sequencing. We can attach sequencing adapters, and then we can give it to the sequencer. So library preparations, they involve preparation of the fragments for sequencing. So that involves purification, adapter ligation, screening, and we can have single reads, or we can also have the paired end reads.